Chromosomes. In this section, we aim to describe the structure of prokaryotic chromosomes and identify the key differences between prokaryotic and eukaryotic chromosomes. These differences include how cells divide, how DNA is compacted, and the presence of structures found only on linear chromosomes. Understanding these distinctions is foundational to grasping the organization and behavior of genomes across the domains of life. To fit massive DNA molecules inside cells, DNA interacts with specific proteins to form a condensed structure called chromatin. Chromatin varies significantly across the domains of life. Bacteria usually have circular DNA, eukaryotes typically have linear DNA with histones, and archaea display a diverse array of chromatin structures. E. coli DNA is a closed circular molecule, approximately 1,600 micrometers long, tightly packed into a cell only 1 micrometer in length. The DNA is organized into a condensed complex known as the nucleoid, consisting of supercoiled and relaxed loops. Current models suggest these loops are anchored to a protein core. Several DNA-binding proteins assist in DNA compaction within bacterial cells. MUC helps organize and compact the DNA, while HNS proteins form bridges across DNA segments. Additionally, free nucleoid-associated proteins contribute by bending the DNA. Together, these proteins ensure that bacterial DNA is efficiently packaged and structurally maintained. In eukaryotes, DNA is packaged into highly condensed structures called chromosomes. Each chromosome consists of a DNA molecule. Reproductive or germ cells have a specific number of chromosomes, referred to as the haploid number, while somatic cells are diploid, containing 2 homologous sets. Chromosomes become distinctly visible only during cell division. When cells are not dividing, we refer to their DNA as chromatin. During interphase, chromatin exists in 2 primary forms. Euchromatin is less condensed and actively transcribed, allowing genes to be expressed. In contrast, heterochromatin is more condensed, often found near the nuclear membrane or at specialized regions like centromeres and telomeres. These regions are generally not transcriptionally active, indicating a role in structural support and gene silencing. This diagram depicts the cell cycle in a typical mammalian cell. It includes the G1 phase, S phase for DNA replication, G2 for preparation, and mitosis. G1 is the most variable in duration, while the other phases tend to remain consistent. Mitosis is divided into 4 stages. During prophase, chromosomes condense. In metaphase, they align at the center of the cell. During anaphase, sister chromatids are pulled apart, and in telophase, new nuclear membranes form around each set. These stages ensure equal chromosome distribution between the 2 daughter cells. Mitosis occurs in somatic cells and results in 2 daughter cells with the same diploid chromosome number as the parent. Meiosis, on the other hand, happens in the precursors of reproductive cells and involves 2 consecutive divisions, ultimately producing 4 haploid gametes. It ensures genetic diversity and reduces the chromosome number by half. The mitotic process begins with prophase, where chromatin condenses and the nucleolus disappears. In metaphase, spindle fibers attach to the chromosomes at the kinetochore. Anaphase follows with the separation of sister chromatids, and in telophase, the nuclear envelope reforms, and chromosomes decondense. The cell completes division during cytokinesis. During prophase, chromatin condenses into visible chromosomes, and sister chromatids are held together by the cohesin complex. The nucleolus disappears, and the mitotic spindle, composed of microtubules, begins to form. This structural setup is crucial for accurate chromosome movement in later stages of mitosis. In metaphase, spindle fibers connect to the kinetochores located at the centromeres of each chromosome. The chromosomes are aligned at the cell's equatorial plane, preparing for equal distribution. This precise arrangement ensures that each daughter cell receives an identical set of chromosomes. Anaphase begins when cohesin proteins are cleaved, allowing the sister chromatids to separate. Now considered individual chromosomes, they are pulled toward opposite poles of the cell by the shortening of spindle fibers. This movement is essential for balanced genetic distribution. In telophase, the spindle fibers disassemble and nuclear envelopes reform around the separated chromosome sets. Nucleoli reappear, and the chromosomes begin to decondense into chromatin. Cytokinesis often follows, resulting in the formation of two distinct daughter cells. Meiosis is a specialized form of cell division that reduces the chromosome number from diploid to haploid, producing gametes such as sperm and eggs. It consists of two successive nuclear divisions, meiosis I and meiosis II. The first division separates homologous chromosomes, and the second separates sister chromatids, resulting in four genetically distinct haploid cells. In prophase I of meiosis, chromatin begins to condense, and homologous chromosomes pair up in a process called synapsis. These fully paired chromosomes are known as bivalents or tetrads. At this stage, crossing over occurs, resulting in chiasmata, cross-shaped structures where homologous chromosomes exchange genetic material. This genetic recombination is crucial for creating genetic diversity in gametes. During metaphase I, spindle fibers attach to the paired homologous chromosomes and align them at the metaphase plate. In anaphase I, the homologous chromosomes are pulled apart toward opposite poles of the cell. Telophase I follows with spindle disassembly and the formation of two new nuclei, often blending seamlessly into the second meiotic division. The second meiotic division resembles mitosis. In prophase II, chromosomes start moving toward the midpoint of the cell. During metaphase II, they align along the metaphase plate. In anaphase II, centromeres split and chromatids, now considered individual chromosomes, migrate to opposite poles. Finally, in telophase II, the chromosomes decondense and nuclear membranes reform, completing the formation of haploid cells. Chromosomes can be stained to reveal distinct, reproducible banding patterns, allowing them to be identified and assigned numbers. They are ordered from largest to smallest. Each chromosome consists of two arms, a short arm called P and a long arm called Q. Bands on the chromosomes are numbered outward from the centromere, enabling precise genetic mapping and analysis. A karyotype is a visual arrangement of an individual's chromosomes, ordered by size and structure. Humans have 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes. This visualization is commonly used in genetic diagnostics and is especially helpful in identifying chromosomal abnormalities. Advanced techniques such as fluorescent in situ hybridization FISH and spectral karyotyping enhance chromosome visualization. These tools allow scientists to highlight specific DNA sequences and rearrange chromosomes to show homologous pairs. Such imaging provides greater clarity and insight into chromosomal structure and abnormalities. DNA within the nucleus is packed into nucleosomes, the fundamental unit of chromatin structure. The first level of this organization involves interactions between DNA and 5 major classes of histone proteins. These proteins are rich in basic amino acids like lysine and arginine, which give them a positive charge. This charge facilitates binding to the negatively charged DNA backbone. Histones are attracted electrostatically to the negatively charged phosphates in DNA, and this interaction can be disrupted by high salt concentrations. When purifying DNA and histones are mixed in vitro, they reassemble into chromatin, confirming the fundamental role of histones in DNA packaging. Under electron microscopy, uncondensed chromatin appears as beads on a string, where each bead is a nucleosome, a DNA strand wrapped around an octamer of histone proteins. In this segment, we introduce a video that visualizes the structure and dynamics of nucleosomes. You'll observe how DNA wraps around histone proteins, how the structure is stabilized, and how higher-order packaging contributes to chromatin architecture. Chromatin is packaged at multiple levels. The first level of organization is the nucleosome, which involves DNA wrapped around histone proteins. There are 5 major types of histones, all rich in the basic amino acids lysine and arginine, both positively charged, which allows interaction with the negatively charged phosphate backbone of DNA. Each nucleosome consists of about 146 base pairs of DNA wrapped around a core of 8 histone proteins, 2 copies each of H2A, H2B, H3, and H4. The DNA is negatively supercoiled, with AT-rich minor grooves often facing inward toward the histones, allowing tight bending. These interactions are stabilized by hydrogen bonds. Histones are electrostatically attracted to DNA. This attraction can be reversed using high salt concentrations. Experiments show that purifying DNA and histones can self-assemble into chromatin, indicating their compatibility. Under the microscope, uncondensed chromatin resembles beads on a string, each bead representing a nucleosome. Condensins and topoisomerase 2 play vital roles in organizing and stabilizing condensed chromosomes. Topoisomerase 2 helps untangle DNA between chromosomes, while condensin I can introduce positive supercoils in vitro. Condensin 2's function is less well understood. Both appear along the long axis of the chromatid. According to the scaffold model, non-histone proteins form a central axis or scaffold, and DNA loops out from this structure. Histone-depleted chromosomes in experiments still show this scaffold organization, indicating it may be a stable architectural framework for chromosome compaction. This illustration shows the scaffold structure and some of the surrounding DNA loops. It summarizes the concept of genome packaging. DNA is first organized into nucleosome arrays, then differentially packaged into euchromatin and heterochromatin depending on gene activity. This figure also summarizes chromatin compaction. Linear DNA is wrapped around histones to form nucleosomes. These organize into chromatin fibers, which are further folded and looped into condensed metaphase chromosomes, visible during cell division. The centromere is the chromosome region where spindle microtubules attach during cell division. It's also the last connection point between sister chromatids. Centromeric DNA contains highly repetitive sequences, and specialized proteins assemble there to form the kinetochore. In the current structural model, the chromatin of the centromere folds into a cylindrical coil that supports kinetochore formation. This three-dimensional organization facilitates the spatial arrangement of kinetochore complexes and enhances mechanical stability during mitotic spindle pulling. This specialized folding is distinct from the rest of the chromosome and plays a key role in how chromosomes resist tension and ensure fidelity in segregation. This model highlights the three-dimensional organization of the human centromere. Chromatin fibers in this region are believed to fold into a cylindrical coil, which helps recruit and organize the kinetochore complex for proper chromosome segregation. Telomeres are protective caps at the ends of linear chromosomes, made up of G-rich sequences and a single-stranded 3 overhang. This overhang can range from hundreds of bases in yeast to thousands in vertebrates. To prevent the ends of chromosomes from being mistaken as DNA breaks, the telomere folds back on itself to form a protective loop, effectively shielding the chromosome tip from degradation and unwanted repair. This looping structure forms what's known as T-loops and D-loops, where the single-stranded overhang invades a duplex region of telomeric DNA. These structures are stabilized by a set of telomere-binding proteins, creating a cap-like architecture that prevents chromosome end fusion and degradation. This spatial configuration is critical for preserving chromosome integrity over successive cell divisions. Telomeres are maintained by the enzyme telomerase, which extends the 3 ends to counteract progressive shortening during DNA replication. In diseases such as Werner syndrome, mutations in helicase enzymes involved in telomerase activity can cause premature aging. In contrast, cancer cells often reactivate telomerase, allowing for limitless divisions. Thus, the balance of telomere maintenance is central to aging, cell proliferation, and genomic stability.